All right, everyone. Thank you very much for coming to PNRI Science Matters seminar series. This week, we're really privileged to have Dr. Kathleen Burns from the Dana-Farber Cancer Institute. Dr. Burns earned her MD and PhD from Baylor College of Medicine, then completed a residency and fellowship at Johns Hopkins, where she also started her lab. And in 2020, she was recruited to Dana-Farber, where she's currently chair of the Department of Pathology. Kathy's early work really brought this new understanding of understanding variation of transposable elements, not just amongst people within populations, but also somatically within individuals. And more recently, Dr. Burns's lab has begun to focus on understanding this really fascinating dysregulation of LINE-1s and other transposable elements in cancer, including understanding how LINE-1s might contribute to cancer progression and evolution, how they might be used as biomarkers, and ultimately how they might be used as effective therapeutics. So in addition to this really impactful body of work, Kathy has always been really one of the kindest and most welcoming leaders in the TE community, where there are a few, kind of grumpier, older leaders. But she's really played a key role in organizing tons of meetings and conferences. And she's always been the one to be there with this welcoming, encouraging face to mentor young scientists, some of them who are just starting their sort of insatiable obsession with TEs. So we're really excited to have Dr. Burns here, and I'll turn the podium over to her.
Thank you so much for that introduction. It's one of the nicest things I think a scientist can say to another is that they're welcoming and encouraging of people coming into the field. I do think we're at a wonderful time right now in terms of transposable element biology, finding many rich intersections, interfaces with other disciplines in medicine. And our group has been really focused at that intersection with biomedical research. And as you said, with cancer research, where I think there's a lot of promise now in the field because of the people working in the field and the intrinsic biology. So very excited to share it today with this audience. I will be giving a talk primarily on our work on LINE-1, and I've titled this presentation, A Retrotransposon in Cancer, the Marker and the Mutator, referring to the two proteins encoded by the LINE-1 element. So this audience may need very little introduction to the fact that much of our genetic makeup is interspersed repetitive sequences owed to the activity of transposable elements, self-propagating elements in our genome. A lot of that activity is resident in the so-called long interspersed element population, or the line elements. Those are the only active protein coding mobile genetic elements today in humans. And so I'll focus a lot of my time on LINE-1, and in fact, a small subset of those elements that have been active in our species specifically over the past few million years. The line reverse transcriptase, though, has added enormous swaths to our genome over evolutionary time. You mentioned their contributions to genetic variation in human populations, where they are a rich source of genetic diversity. That's because of line making copies of itself in the genome. But also line can retrotranspose in trans, so-called short interspersed elements or signs, ALU sequences, which have really undergone a burst of retrotransposition and expansion in primate lineages and continue to be very active in the human germline. There is by and large less sign retrotransposition in human malignancies, and I won't focus so much on those sequences today. We have other so-called RNA transposable elements or copy and paste elements in our genome derived from viral sequences, the LTR elements. Again, fascinating biology there, certainly from a regulatory perspective, although I won't touch on them much today. And I won't describe at all the DNA.
Did you have a car accident, Lloyd? Yeah, that's
I won't talk at all about DNA transposable elements, the so-called cut and paste elements. Those are dead for activity in the genome as transposons, however, have been co-opted in really fascinating ways. For example, the recombination machinery responsible for VDJ recombination in B lymphocytes and T lymphocytes are derived from DNA transposons. This is all making up, on the order of half of our genome. Um, Meanwhile, unique protein coding exons in our genome make up a much smaller slice of our genetic makeup, 1% or so of our DNA. And most cancer genetic studies have been really laser focused on those relatively unique sequences. Our lab has always been driven by our curiosity for the relatively understudied parts of our genome. So with that backdrop, I'll see if I can advance a slide here. Go. A little bit about LINE-1 biology is shown on this slide. This is how LINE-1 makes copies of itself in the genome. Our understanding of this process is that most LINE-1 elements are fragments of long ago active sequences with degenerating open reading frames, or 5' truncated sequences on arrival not capable of coding for proteins. But each of us inherits on the order of 100 or so potentially retrotranspositionally competent elements, meaning they have intact promoter sequences and intact open reading frames. And our thinking is that most of the time these elements are kept under control in cells through multi-layered defense mechanisms, partly reliant on DNA methylation. So promoter methylation can silence the internal RNA pol-2 promoter of the line element. But when that silencing mechanism is something that's not completely secure, as we think happens in many human cancers, you can have production of two proteins from a bicistronic transcript. These are 6 kilobase long transcripts encoding 2 open reading frames, proteins essential for LINE-1 to retrotranspose or make a copy of itself in the genome. The first of these proteins, for our purposes today, we will treat as our cancer marker, the open reading frame 1 protein. Mechanistically, what it does in LINE-1 retrotransposition is still not very well understood. It is essential for retrotransposition. It forms a homotrimeric complex and possibly higher order complexes, and is an RNA binding protein, very important in the assembly of this RNP with the ORF2 protein and the line RNA that encoded it. The ORF2 protein, from a mechanistic standpoint, we have a bit of a better handle on. It has 2 very obvious domains that are critical for retrotransposition. One is the endonuclease domain. This is related to the APE endonuclease. The other domain, a reverse transcriptase. And those domains act in a coordinated fashion to execute new integrations going into the genome. The endonuclease nicking our genome at a location, as I'll describe, has a preference for a short stretch of A's and T's. And then those T's from a 3' hydroxy nick that's liberated from the genome can base pair bind with the poly A tail of the line RNA. That's a setup for target primed reverse transcription by the line encoded reverse transcriptase, also there resident in the second open reading frame protein. That gives a cDNA copy of the line that encoded it. We know that this is how you get new insertions of line in the genome, although exactly the steps to resolve this intermediate structure and any host factors involved remain only partially understood. It also is increasingly clear that you can also have other untoward outcomes of this besides a new insertion going into the genome. There's a potential here to have these result in DNA breaks and more complex rearrangements and DNA damage. The ORF2 protein, therefore, being a mutator, we think potentially of some importance in cancer genomes. Rick mentioned that I started my research group at Johns Hopkins. It was a wonderful place to be a physician scientist and become interested in line 1 elements. It was really the place where Haig Kazazian and colleagues first recognized that there were active mobile genetic elements, active line 1 sequences in humans. And that was a study published in the late 80s. Where they surveyed for genetic causes of hemophilia and found 2 independent instances where the factor VIII gene mutation responsible for this coagulopathy were caused by line insertions into, critical exon of this gene, very clearly producing deleterious mutations. And when Haig first wrote about this, he wrote in the manuscript that insertion of line elements, L1 elements, involving retrotransposition of DNA sequences through an RNA intermediate into a new and distant location in the genome represents a mechanism for mutation to produce human disease fundamentally different from those previously described. We really view this as like a prescient statement in this paper and continue to be very interested in line activity as a special type of mutagenic process. So with that backdrop, I'll go a little bit deeper now and tell you more first about the first of these line-encoded proteins. Again, for our purposes today, a cancer biomarker. So the structure of open reading frame 1 protein has been worked out in some elegant papers by Oliver Reichenreiter's laboratory at the University of Tübingen in Germany. Oliver has, from those predictions, suggested that open reading frame 1 protein forms a homotrimeric RNA binding complex through interactions between its coiled coil domains across molecules. He's also proposed that those coiled coil domains may fold open. There's certainly a flexible sort of hinge region in this kind of almost end-terminal segment of the protein. And he's proposed that these may create higher order lattices in cells. There's some additional work from NYU and other investigators suggesting that open reading frame 1 protein can form sort of phase separated RNPs in cells. Um, it was well known as we were getting started in the field that open reading frame 1 protein was highly expressed in, for example, germ cell tumors, embryonal carcinomas, but really the extent of its overexpression in cancers wasn't that well appreciated until our group and now others have come on the scene with monoclonal antibodies to really facilitate detection of this line-encoded protein. Our early studies on open reading frame 1, done in collaboration with a large group of pathologists at the Johns Hopkins Hospital, led by Nemanja Radic, who was a pathology resident in our lab at the time, really underscored that there was broad expression of open reading frame 1 protein in some of the most commonly occurring epithelial malignancies, particularly P53 mutated cancers. This is true in esophageal cancers, gastric cancers, small bowel and colon cancers. Here's a nice photo micrograph that shows you brown staining of open reading frame 1 protein accumulating in these nests of colon cancer cells, whereas histologically normal adjacent colonic epithelium has much less accumulation of this protein. It's also seen outside of the gastrointestinal tract in head and neck cancers, in lung cancers, in breast cancers, prostate cancers, ovarian cancers. So very much, as we have said, a hallmark of malignancy is that there is this overexpression of open reading frame 1 protein. There's also beautiful evidence that antedated our studies and that have emerged since that the line promoters can be hypomethylated in many of these malignancies, and that line can act as a somatic mutagen in many of these cancers. In some cancer cases, line induction really appears to be essentially a binary switch, a signal of the cellular transformed or cancerous state. And probably the best example of that is in precursors of high-grade serous ovarian cancer. High-grade serous ovarian cancer, sort of a misnomer ovarian cancer. This disease actually appears to arise in the fallopian tubes, and it's the luminal epithelial cells of the fallopian tube that are thought to really seed these malignancies. These malignancies have very complex karyotypes and almost universally have p53 mutations that are accumulated in the precursor lesions and carried forward to the fulminant cancers. These fallopian tube cells normally do not express open reading frame 1 protein. This is work that we did in collaboration with Yiming Shi's lab at Hopkins. Tom Pasanik led a lot of these studies. So open reading frame 1 protein is virtually absent in normal fallopian tube epithelium. And Tom and Yiming, as they began to look at the so-called stick lesions, precursors of high-grade serous ovarian cancer, shown here at the same magnification, you can see how different these cells look. They're much larger, have open chromatin, very large nuclei. They're beginning to fix p53 mutations and become chromosomally unstable. These cells accumulate mutated p53 in their nucleus, as you can see by this immunostain for p53 protein, and perfectly coincident with that in time and space, there's an induction of open reading frame 1 protein accumulation in the same cells, indicating really robust expression of this retro element. Rick mentioned that we are now interested in using open reading frame 1 protein and other potential biomarkers coming from the repetitive genome to change the way we diagnose cancers. And he's exactly right. Of course, our hope in doing that would be that we would be able to detect cancers not just with a tissue sample in front of us, but ideally in less invasive ways. So we went looking for the protein, for open reading frame 1 protein in the peripheral blood of cancer patients. I started these studies initially working closely with Marty Taylor at the time at Johns Hopkins. And Marty later moved to Boston, to the Massachusetts General Hospital for his pathology residency and struck up a fantastic collaboration with David Walt's lab and a postdoc fellow there, Connie Wu, who really helped us to break a barrier of the lower limit of detection of this protein in the blood. So standard sandwich ELISA assays will not allow you to detect this analyte in the peripheral blood of cancer patients. It's really in trace quantities in the peripheral blood. But Connie and David Walt's lab have really focused on developing ultrasensitive single molecule protein detection strategies, including the Simoa platform that's illustrated here. In this case, the principle of the assay is similar to a sandwich ELISA. Our analyte open reading frame 1 protein is basically captured by two affinity reagents, one conjugating it, one conjugated to a bead, in this case, a camelid nanobody that we made with an affinity for open reading frame 1 protein. And then you can come in with a detection reagent that provides an enzyme for a chemical reaction, then giving you the ability to detect this example as an on bead with the analyte of interest and to distinguish that from off beads where there is no analyte. What is important about the Simoa platform is that the signal is not diffused in a very large volume, but that individual beads are segregated to very small microwells so that you get this kind of digital readout of on beads and off beads. This really lowers the lower limit of detection of this analyte and makes it possible to detect in the peripheral blood. Still, even with these very ultrasensitive strategies, in most people, it's not possible to detect open reading frame 1 protein in their peripheral blood. So here we look at more than 400 healthy control samples, and you can see that open reading frame 1 protein levels, this is now using peripheral blood, small volumes of peripheral blood. Open reading frame 1 protein is not detectable in almost any of the samples. Just about 1% of the time we have exceptions. I'll come back to this one individual who has very high levels. You can see, though, as we went to different collaborators and collected samples of patients with advanced cancers of different types, we see a very different landscape. And Samoa assays reveal the presence of open reading frame 1 protein in the peripheral blood of these individuals. It's most commonly detectable in ovarian cancer patients. That's not a surprise, and I'll show you a bit more data to substantiate that in an independent cohort. We can also see expression in colon cancer and to lesser degrees in lung cancers, other types of cancers. That all fits very nicely with our immunostaining data about this marker. I said I would come back to this one individual from our healthy cohort. At the time that this blood sample was collected, they were thought to be a healthy individual. Later, however, shortly after this sample was taken, the person was diagnosed with a metastatic prostate cancer as well as a cutaneous T-cell lymphoma. These could be sources of the open reading frame 1 protein in this control sample. We have iterated a bit on this assay. There's still like pretty new assays in our hands. You can read more about them in our recent cancer discovery paper describing our strategy for continuing to improve upon these assays. In that manuscript, we've described second generation Samoa assays, for example, as well as other flow cytometry based assays, the mosaic assays, which allow us to put through a flow cytometer a number of these beads and detect very, very trace quantities of open reading frame 1 protein. One of the real technical advances really has been the development of a bunch of great affinity reagents against the protein. So if we began to understand tissue expression because of our production of a mouse monoclonal antibody against open reading frame 1 protein, really these ultrasensitive assays have been enabled by a suite of rabbit monoclonal antibodies developed in our lab at the Dana-Farber, as well as camelid nanobodies that have been developed and engineered in collaboration with John LaCava and Mike Rout at the Rockefeller. In this particular slide, I'm showing you the performance of these different Samoa assays in a different cohort of ovarian cancer patients and healthy age-matched control women. These samples were given to us by Ronnie Dropkin at the University of Pennsylvania. And you can see that depending on the assay and its performance characteristics, we're able to detect open reading frame 1 protein in various kind of quantities of the patients with ovarian cancer. And in most cases, we can draw a pretty clear threshold, even in our most sensitive assays where we may be detecting some of this protein in healthy individuals, a pretty clean threshold between those individuals and individuals with cancer. Still a lot of work to go to understand the significance of these types of findings and what their clinical utility may be. One thing that we did in this particular pilot experiment was to mix in small numbers of women with early stage malignancies, stage 1 and stage 2 ovarian cancers. The disease is relatively localized and more treatable in those early stages. And unfortunately, very few women are diagnosed with early stage disease as compared to much later in the disease development in advanced disseminated cancers when they become symptomatic. So we were encouraged to see the assay doing reasonably well with these small numbers of stage 1 and stage 2 patients, raising the possibility that it may be useful in early detection. I mentioned the Mosaic platform, just another variation on these types of on-bead assays. In this particular setup, there's a fluorescent probe, a very robust signal amplification, and individual beads are counted by flow cytometer assays that makes open reading frame 1 protein detectable in patients with gastroesophageal cancers, for example, where they had not been detectable by the standard Samoa assay. There's a question in the chat, which I'll address as I go along. Thanks for the question. Are there any secondary factors that seem to explain which cancer patients have detectable versus undetectable open reading frame one protein, subtype, tumor location? It's a really great question. At this point, we don't have a great understanding for that. We're just beginning to look now at patient cohorts where we actually have tumor sample as well as matched peripheral blood. So we can ask, is the antigen there in the tumor? And we can, potentially look for other markers that might be an indication of cell death in that tumor. We assume that that's how this intracellular protein is getting to the periphery. Lots still for us to work on. Whether tumor location is a major contributor, we're not sure, or how faithful a burden of disease marker this is. Is it a good detector of bulky malignancies as opposed to less so? We don't know at this point. I have done just a little bit more work to think about uses of this marker clinically. A very important clinical need, of course, is being able to monitor patients over the course of treatment and to potentially detect whether or not there is minimal residual disease after a therapeutic regime, to decide at that point whether you would intensify therapy or not. The strategy has been very important in leukemia treatment and is increasingly something sought after for the management of solid tumor patients. So we took here a small gastroesophageal cohort of patients and looked at their responses to a neoadjuvant chemotherapy and radiation therapy regime. The clinical teams, through imaging studies, divided patients into responders and non-responders, and we had open reading frame one protein measures for patients pre-treatment and post-treatment. We were blinded to the clinical scenario when we tested these samples, but clearly now that we can look at the data, patients who responded robustly to this therapy and had remission by clinical imaging studies and other studies had, in all cases, open reading frame one protein drop to undetectable levels from measurable levels in these first generation Simoa assays. Meanwhile, people who had primary refractory disease who did not respond to the treatment continued to have, in most cases, persistent, in some cases increasing open reading frame one protein levels. A reviewer of our paper, and this speaks again to the question in the chat, said, I think there might be a difference even at the pre-treatment time point between your responders and non-responders. So in revisions for this paper, we did in fact break the two groups out. And the reviewer is correct. It does look like there's a bit of a difference even at the get-go. And so that raises the possibility that this could be a prognostic marker as well. Still more work to do to understand really what this means. Could this represent bulky tumor, genetically heterogeneous tumor with greater access to the blood supply? Why is it that those patients do a bit worse? Is something we would like to better understand. So at this point, I hope I've captured your imagination a bit, that the junk genome is in fact dynamic and mobile, at least over evolutionary time, and that there are elements there that then become protein coding in human malignancies and maybe interesting biomarkers of disease in that context. Now I want to pivot to really what the retro element has evolved to do, and that is to reintegrate itself in the genome, to mutate genomes. We think this is potentially a very interesting aspect of this epiphenomenon that LINE is overexpressed in cancers, that in fact it unleashes a mutator phenotype that focuses us now on the second open reading frame protein, ORF2p. We have wanted recently, as I moved from Hopkins to the Farber, to better understand what's the true genomic impact of Line-1 in cancers. In all of the cancer samples that I've alluded to, we now have known for several years that Line is mutagenizing genomes by creating surgical insertions of the Line retroelement into genomic DNA. However, we think that this is really capturing only a small amount of its DNA damaging potential. And to really go after the broader scope of the total genomic impact of Line-1, we have set up inducible experimental systems to model this activation of lines in cancer, and then leverage whole genome sequencing approaches to begin to appreciate its impact on genomes. And I want to say at the outset that this has been driven really in close partnership with a data scientist and assistant professor here at the Dana-Farber, Chengzhang Zhang, who really led all of the genomic sequence analyses in collaboration with our experimental group, as well as Alice Lee's lab here at Boston Children's. So a fantastic collaboration. In my group, Carlos Mendez Durantes and post-baccalaureates Phil Schofield and Jupiter Kalinowski really led the experimental biology work, and Jennifer Carlo from my group participated very closely with she from the Lee Lab and Cheng Zhang in his group on the analytical work. So Carlos set up these cells. In these particular experiments, I'll show you data from retinal pigment epithelial cells, or RPE cells, compromised for p53. That's the only way we can do experiments where we're inducing line. He engineered the cells to have an inducible line-1 system, in this case, a TET-on system, and have a whole series of different types of controls. This particular diagram shows you a DMSO control versus doxycycline used to turn on line. And then Carlos and his team did single cell sorts of controls and Line-1 expressing cells to take then the single cells into whole genome sequencing, or to see how genomic damage evolves over time. He's grown out clones for whole genome sequencing. This has been really impressive to me in terms of it underscoring how Lyme can incite structural chromosomal instability. So these graphs just capture at a really high level kind of the overall landscape of copy number alterations in these cells as we appreciated by these whole genome sequencing studies. So you can see in DMSO treated cells in this kind of control group, again, p53 compromised backgrounds, that sometimes about 15 or so percent of single cells have a few de novo or acquired copy number alterations. They don't have really large numbers for the most part of these. We sometimes do, though, see terminal deletions naturally occurring in these cells. When we turn on Line-1 and force its expression with a doxycycline inducible system, that proportion of cells with copy number alterations increases. And we also see the emergence of cells with much more complex alterations. So cells with multiple, in some cases, tens and scores of copy number alterations. In both groups, one of the most common types of alterations are terminal losses. You can see how those increase in frequency with line expression. We can also more uniquely see different gains and complex rearrangements or copy number alterations in these cells with line expression. When we allow the single cells to grow out into clones, clonogenicity, interestingly, goes way down. So line expression is actually genotoxic to a degree in these types of assays. If we set the controls at 100% clonogenicity, fewer than half of the cells with a history of line exposure are able to grow out a clone. Those that do, again, have copy number alterations that persist. You can see in a much higher frequency of these copy number alterations, 1, 2, 3, 4, and sometimes more copy number alterations. You'll notice by and large the numbers are lower than with the single cell analyses. We presume that the cells with the most damage never clone and grow out for these longer term experiments. Again, you can see terminal losses persist in these cells. In some cases, whole chromosome losses. What evolves over time are more subclonal features, losses that evolve over time, and I'll talk a little bit more about that evolution of chromosomal instability on the next few slides. So first, just to show you a bit of an example or two of these instances, we recently posted a bioarchive paper where you can read more about the details of these and other rearrangements. In this particular sample, we have a clonal outgrowth of a doxycycline induced cell. There is a endonuclease cut site for the line-1 at a place where we have lost one end of an arm of the short arm of chromosome 9. And you can see that that's affected copy number along the whole chromosome, so likely leading to some instability of that, that copy of chromosome 9 in the cell population. Sometimes we see real alterations in genomes, as I said, that go on to evolve. In this particular case, we have a breakpoint on chromosome 5 that leads to terminal loss of that sequence. We can see a retrotransposed pseudogene, so a cellular mRNA that was picked up by ORF2 being overexpressed in these cells, added to one end here, added to the broken end of chromosome 5. It has recombined with, and so there's a continuous sequence with a similarly retrotransposed pseudogene in this case at the end of chromosome 18. And so what we think has happened is that you get fusion of these two ends, these two Q arms of chromosome 5 and chromosome 18, which is something that creates a dicentric chromosome that is obviously an unstable structure. We think as the clone has expanded that you see this adjacent interval of subclonal loss in some, outgrowths of these cells, owing to a series of breakage fusion bridge cycles that have led to the destabilization of this dicentric derivative of chromosomes 5 and 18. So a very interesting, very clearly line-mediated event breaking both chromosomes, placing them together, but it's a fusion that leads to continued breakage and evolution of the chromosome over time. Not all translocations create that sort of instability. Sometimes there are clean, balanced translocations that can result from the overexpression of line. Here's a nice example that basically creates a nicely balanced reciprocal translocation involving chromosomes 16 and 17 in one of our clones. On one of these derivative chromosomes, you can actually see the products of two retrotransposition events are recoded. Line 1 at one end, a retrotransposition reaction attached to the remainder of 16, and another pseudogene integration attached to the 17 fragment that's here. Interestingly, the derivative, the sort of opposite piece of this, the other derivative chromosome does not have retrotransposed sequences there. You just have a new junction between the two chromosomes remaining. we actually quite frequently see breaks like this that we think are really attributable to the line overexpression in the system, but without a signature line 1 insertion there, these types of things, when they occur in cancer genomes, are not, assigned to line 1 sequences, but happen with a lot of frequency in our experimental systems. A little bit more about these inducible systems, just to think about consequences of this type of DNA damage for cell biology. Now I'll take you back in time to a report we published now several years ago, led by Dan Ardelian in my lab at Hopkins. Dan is now a postdoc fellow at the Broad Institute with Todd Golub, a medical oncologist by training. He was working on his PhD with us in Baltimore, and already then thinking like an oncologist, he was really interested in what he perceived to be the paradoxical observations that Lyme could be genotoxic in cell culture systems and yet highly expressed in human malignancies. So he set out to better understand what genes, what cellular pathways are important for cells to survive Line-1 overexpression or cope with retrotransposition. He also used this RPE type of inducible system. In Dan's case, he had a p53 knockdown for many of his studies, although he also has some work in this NSMB paper describing how important it is to lose p53 if you're going to have this type of mutagenic action going on in a cell. Dan used a TET-on system to express now a luciferase transgene as a control, or again, a line construct as our experimental branch of these systems. Dan then did cell fitness screens, basically forward genetic screens, creating pools of cells with a knockout for every gene in the genome, and then grew out these cells over the course of several weeks with serial sampling to look at guide RNA representation. And then, of course, sort of normalized his guide RNA representation so that non-targeting guides were equally sort of represented in the line positive pool and luciferase, which was smaller at this point than the luciferase expressing cells. So what Dan appreciated in doing this is that a series of different pathways in cells become really important if they are sustaining line-associated mutagenesis. And really, one of the top kind of pathways overall, but particularly in thinking about DNA repair pathways that comes out of this, is the Fanconi anemia DNA repair pathway. So Fanconi anemia loss is acceptable in RPE cells, not too damaging to their fitness. You can see these black lines, which represent the growth of knockout cells with the respective guide RNAs in the luciferase controls. You can see that in a luciferase control cell, they can lose Fanconi anemia A, Fanconi M, Fanc-L, D2I, different components of the Fanconi anemia core complex. One of the translocases that identifies lesional DNA, a ligase that monoubiquitinates FANC-D2 and FANC-I, which are involved in the then triggering of a DNA repair pathway. Also, Fanconi anemia S or BRCA1. All of these are well tolerated mutations in the parental cell line, but when Dan turns online, you can see that these guide RNAs do not survive in the line expressing cell pool. Those guide RNAs, again, reflecting the growth of knockout cells for the Fanconi anemia gene, really are being lost from the pool by on the order of 2 weeks, 3 weeks into the screen. So a very striking molecular dependency that's been induced by line overexpression in these cells. Let me try to advance my slide again. Dan did a little bit more work in this follow-up paper to demonstrate that, in fact, line expression is activating the Fanconi anemia complex. I'm just showing you one piece of those data here. He looked at FANC-D2 nuclear foci in cells, in S-phase cells, you can see that untreated cells have some activation of this complex, some nucleation of Fanconi anemia pathway, coordinated DNA repair, and that's increased substantially in cells with hydroxyurea, DNA damaging agents. It's actually pretty comparable to the overexpression of wild type line, and that is mitigated when we instead express a line construct that's mutated for the reverse transcriptase. So interestingly, and many of our DNA damage assays look like this, it doesn't appear that the DNA damage is owed exclusively to the endonuclease activity. It seems there's something about the ORF2p reverse transcriptase, or rather the lesions that are produced by that RT in the context of a line that really are more challenging for these DNA repair pathways. So there are many other data on this topic from our lab and others that I won't have time to review with you today, to underscore the idea that line retrotransposition may create DNA lesions that essentially pose a conflict for replication, and therefore induces the requirement for these replication coupled DNA repair pathways, pathways that are really critical for cells to get through S phase. That includes in Dan's screen synthetic lethal interactions with line expression for all of these components of the Fanconi anemia complex, as I described. Also, components of the replication stress signaling complexes, like non-essential components of the 911 signaling complex, and ATR interacting protein. ATR, of course, itself not being something we can detect in screens like this, as it's an essential gene. Dan also found that replication fork restart factors like the Bloom helicase, Werner helicase, and Werner interacting protein all become critical for line 1 positive cells to grow in culture in these experiments over this period of time. Orthogonal to our work and completely independently, the Wasaka lab at Stanford, the Buka lab at NYU have shown that knockout of the Fanconi anemia complex can enhance the efficiency of line 1 retrotransposition. And we have several experiments in Dan's paper and in Carlos's bioarchive paper showing that loss of these genes can enhance line 1 associated DNA damage. And I've shown you data that line expression can increase FANCD2 nuclear foci in S phase cells. Building a bit on these findings, a postdoc in our lab currently, SN Isik, is doing some work to really directly demonstrate the potential for conflicts between line 1 retrotransposition intermediates or lesions created by line 1 and the progression of DNA replication forks. So she does these studies. Here she's taken a cell line from Carlos, absent P53 with a doxycycline inducible line 1 system. She turns on line at different time points here, and then treats the cells with labeled nucleotides for pretty short pulses, 30-minute pulses in each of 2 colors, and then creates these spreads of DNA fibers on glass slides so that she can look at the progression of replisomes over time. When she sees then a tracked length, she can add red and green colors together and see how much nascent DNA was made in the course of that like hour-long experiment. You can see that in control cells, replication soldiers on with these additions of labeled nucleotides, and she can get quite nice long track lengths in fibers that are well spread on a slide. Meanwhile, when she forces line expression for 3 days, for 5 days, you can see how the tracks really slow down. She's inducing a phenotype that field calls replication stress, meaning that you can't have nice progression of replication forks. Occasionally, she can find fibers that actually capture the first color pulse, the red here, if you like, central to flanking IDU only or green tracks. And that's a pattern that indicates that you had centrally an origin fire during the first labeling phase of this experiment. The two sister forks then progress outward in opposite directions and keep going. And the expectation is that when you then stop the experiment, those sister forks will have had the same period of time to travel, and you should get equal stretches of IDU only sort of green tracks on either side. And exceptions occur when you have sort of asymmetric sister forks, where one of these forks has had time to progress in an unimpeded way, and the other one appears truncated or blocked in these experiments. So the sister fork ratio then really can drop off. And that's in fact exactly what Essen sees when she induces line. It really suggests that there are discrete DNA lesions caused by the expression of the retro element that are halting some of these forks, that there may be a global slowing, but that's because of specifically challenged or stalled sister forks. So she is doing more work with these really elegant systems to try to understand what are key factors in this replication stress phenotype. So with that, I want to stop and save ourselves a few minutes for questions. I hope that this has given you a nice overview of some of our thinking about the LINE-1 transposable element and its role in cancers. Just to sum up, your genome, all of our genomes, is really shaped in a huge way, profoundly, by the activity of transposable elements. And while perhaps for a long time these repetitive sequences have been dismissed as junk DNA, there's some fascinating biology there for us to understand. Of the transposable elements that are so much a part of what makes our species what we are, there is a single really dominant protein coding active retrotransposon, and that's the LINE-1 element. It is not just something that generates diversity in germlines, but is also expressed and active as a mobile genetic element in cancers. It's clear that open reading frame 1 protein and open reading frame 2 protein are expressed then in many malignancies. We think there's opportunity for the first protein because it's so readily detectable. And in fact, with special assays, even detectable in the blood, we think there's tremendous potential to think of this as a cancer biomarker. We have termed it a hallmark of cancer, and we really are interested in developing assays to non-invasively profile patients, potentially the general population. We don't know if the assays will ever be that good, or high-risk patients or patients under treatment, to see if this can be useful for monitoring the emergence of these diseases, or to track these diseases over time. Meanwhile, the second protein encoded by line, open reading frame 2 protein, is a genome mutator. We think it's there, although in trace quantities, wherever you have robust expression of open reading frame 1 protein. We know that it is causing new insertions of LINE-1 in cancer genomes, and we're also thinking more broadly about it as a DNA damaging agent, potentially breaking chromosomes, inducing replication stress, and enforcing in cells dependencies on specific DNA repair pathways for cell growth. So our working model is that Lyme may be contributing in important ways to chromosomal instability in the evolution of cancers. And as Rick alluded to at the beginning, I think this whole paradigm has some implications for how we might think about treating cancers. So many of our effective cancer therapies are, at essence, DNA damaging agents. Our understanding of this endogenous mutagen in these diseases gives us the potential to try to exacerbate its effects, to produce epigenetic dysregulation that promotes the expression of Lyme further in these diseases may push cancer cells past a tipping point. Other tactics to try to increase production of open reading frame 2 protein or to inhibit associated DNA repair pathways may help to harness this type of activity into an effective cancer therapeutic. A very long way away, but has been a really wonderful thing to be thinking about and beginning to work on. So with that, I'll stop and thank some key collaborators and people in my lab who've contributed to these studies over really many years. I've shouted out about several of them along the way. Our open reading frame 1 protein work really began with Nemanja Radic and collaborators in the pathology department at Hopkins. Nemanja worked with several fantastic people there. Jared Starenka in our lab, and he worked on somatic retrotransposition in cancers as well. The mantle now has been taken up by our collaboration with David Walt's lab. That was really spearheaded by Marty Taylor, transiting between Hopkins and Harvard as well, and teaming up with Connie Wu in the Walt lab for these studies. I mentioned Mike Rout and John LaCava. They've generated the best nanobody reagents that we have for ORF1 binding and have really promoted those biomarker studies. When it comes to the DNA damage work, again, many people in my lab have worked on this over a period of time. Dan Ardillian did a lot of the synthetic lethal screens that I described for you today. Essen is studying the replication stress phenotypes. Carlos, Jennifer, Jupiter, Phil in my lab have really done a lot to set up experimental systems and analytical pipelines for our whole genome sequencing studies. And we wouldn't have had the guts to undertake those types of analyses without a phenomenal collaborator in CZ Zhang. So with that, I will wrap up. Thanks so much.
Thanks very much, Kathy. That was awesome. Do you have any questions from the audience? Maybe I'll start while they're getting their bravery. So this replication fork asymmetry, do you think that's caused by sort of the nucleotide composition or secondary structure of the L1 itself, or you think it's something about the break or the lesion that's happening?
That's a great question. Right now, we don't really know. I think you could potentially challenge a fork through any number of mechanisms. It could be that ORF2 is binding to some sites in the genome and does little from there. Although the RT dependence really makes me think that in many cases, the RT dependence of many of these DNA damaging phenotypes makes me think that in many cases, you may get a sort of intermediate of retrotransposition that's not well resolved. And it's possible that it's those types of structures. We don't know how many there might be in a cancer cell that create barriers to DNA replication and promote break-ins. It's also possible that DNA is just broken and that you have double-stranded breaks, that ORF2 and even reverse transcriptase products are no longer really at the site, and that creates DNA damage. Something we would really like to do now is try to detect intermediates in cells. Just to give you a sense of the numbers, our experimental systems really suggest that for every canonical line insertion that we get, there's another DNA break that is resolved in an abnormal way. And so if you extrapolate, there are many examples of tumor studies now where people know that there are hundreds or thousands of somatically acquired insertions in cancers. That's probably even an underestimate from not targeted whole genome sequencing studies and looking for canonical insertions. That may mean that there are many breaks, and then the evolution of that DNA damage through successive breakage fusion bridge cycles in some cases could really add up and compound to a lot of chromosomal instability. But exactly what the critical lesions are and how cells clear them, we're still trying to get a handle on.
Great, thank you. Michael, did you have a question?
let's see. Yeah. So my question was wondering, so looking at the mechanism of some of these copy number variations, are there line one specific endonuclease inhibitor drugs that could be used to test whether these copy number variations are due to that catalytic activity of the endonuclease, and or due to some other mechanisms like recombination between repetitive elements or other things, like the capture of these other double strand breaks that you're talking about.
Yeah,
I
these are
know
great
in
questions.
yeah, I know in in the retrovirus community, there's a lot of sort of retroviral specific drugs that have been used to sort of block protease, block uh, reverse transcriptase and things. I don't know what there is for specifically for L1s.
Yeah, it's a great question. There's actually a lot of activity in the biotech kind of space now to develop inhibitors that might be more line specific. Certainly, line benefits from some of the virology research that you're talking about because nucleoside analogs that were really designed to inhibit the HIV reverse transcriptase actually will inhibit all kinds of RTs and other error-prone polymerases. So we have good small molecule line inhibitors. It's a really important experiment, and we haven't done that yet to see whether that really mitigates DNA damage in the system. Those drugs can be very effective for stopping line insertions and retrotransposition, but that's not exactly your question, and I agree with you. You're asking the right question. As far as endonuclease inhibitors, there are some groups working on this, but probably we're less mature as a field in terms of thinking about drugs that might be good line EN inhibitors. It's a really great question.
Thanks.
Other questions for Kathy? I don't know that this is a question as much as an interesting thing you said about how you think the therapeutic approach might be activating and encouraging more activity from the line ones as opposed to inhibiting them. Can you say a little bit more how you imagine that might have anti-cancer effects?
Yeah, I'm completely speculating here, but I think people have been interested in line biology in kind of different contexts. There are groups thinking about what I presume are lower basal levels of line activity that may exist in normal tissues. And they're thinking about the reverse transcriptase and possibly other activities of the line as something that may promote innate immune signaling, inflammatory conditions. If that's kind of your model for line action in a biological system, it makes all the sense in the world to me that you might want to inhibit its enzymatic activities and that strategies to inhibit it might be quite effective. Um, Even if Lyme plays a starting role in tumorigenesis by creating some chromosomal instability, my sense is that by the time that's happened, by the time you have a fully transformed malignancy, um, you're past any point where you could ever undo that chromosomal instability or that cellular phenotype by inhibiting line. I assume, but I'm not sure, that inhibiting line at that stage would be pretty ineffectual. On the other hand, the way most of our therapies work is by damaging DNA. That's the basis for so many chemotherapies, and we think for radiation therapy. It seems to me that you may have a therapeutic window in that cancers have a lot of this activity, and even they, with their P53 mutations, can't perhaps withstand endless amounts of this activity. Meanwhile, a normal cell with epigenetic mechanisms intact may be okay if we came up with strategies to leverage line-associated DNA damage. You could imagine enhancing that by compromising epigenetic inhibitors of line expression. You could imagine although it's harder to know the mechanisms to attack, that if you produce more line protein or if it acted longer in cells, that might be a strategy to enhance associated DNA damage. Another potentially more tractable strategy would be inhibiting key repair mechanisms. There's a wonderful field now, I think, emerging in DNA damage response inhibitors, and maybe using those in conjunction with epigenetic agents or using those in a selective way in line high tumors will be especially helpful. A lot still to work out, but that's kind of our framework for thinking about it.
Yeah, that's fascinating. Thank you. Other questions for Kathy? All right, and thank you very, very much for taking the time to be here. We really appreciate it. And to the audience, thank you for joining us, and please join us again on March 19th. Richard Gibbs will be joining us, and thank you again.
Thanks so much. I really appreciate everyone's questions and comments and your time today. Thank you.